All right, well, hello everyone and welcome to today's webinar. I'm part of FlowJo University. My name is Veronica Obregón-Perco and I'm an application scientist with BD offering support for FlowJo and SeqGeek. And so today's webinar, we're going to be talking about an introduction to our single-cell RNA sequencing platform, or SeqGeek version 1.7, in case you're following along with the download. So a couple of housekeeping things for today's webinar. Firstly, it will be recorded, and I'll show you at the end of my slides where you can access that video from today, as well as a library of some of our previously recorded webinars as well. If you have any questions at any point during the webinar, don't hesitate to ask. You don't have to wait until the end. If you have a question, you don't have the ability to unmute and ask it out loud, but you can go ahead and post it in the chat, and I'll try to monitor that throughout the slides and try to answer them in real time if I can. Also, you can feel free to email me at the end of this webinar or after watching this recording if you have any follow-up questions. So here's my email. Pretty easy to remember, just my first name and my last name at bd.com. So feel free to reach out if anything is unclear during the session today. And so in terms of format, I'm going to go ahead and present maybe 20-30 minutes worth of slides just to kind of introduce the topics and make sure we're all on the same page with the verbiage for the software. And then the rest of the time I'll spend in the software really showing you the point and click execution of how to get started with your own analysis in the software. All right, so I'm going to go ahead and get started. And I'm going to go ahead and also stop my video feed just to make sure my internet can keep up with us today. All right. Okay. So with single-cell RNA sequencing, we can get a lot of information, definitely more granularity than we would get with traditional bulk sequencing, and also more depth than we could get with traditional flow cytometry, at least with current technologies. But I think with single-cell RNA sequencing data comes kind of the task of having to get in there and get meaningful information out of it. And I think that's where a lot of holdup often happens is once we invest money into designing an experiment and doing a single-cell RNA sequencing run, we end up with just all this data, and it's a little overwhelming on how to get started with it if you're not a bioinformatician or not really a trained data scientist to deal with such large data sizes. So what is out there for just the run-of-the-mill bench scientist who doesn't really have those skills, at least at the moment, but wants to try and tackle the data they've gotten from a single-cell RNA sequencing experiment. So this is really how SeqGeek came about. It was kind of the flow cytometrist approach on how to tackle single-cell RNA sequencing data. So why use SeqGeek over, say, other methods of digging into bioinformatic data? Well, as I'm alluding to, it doesn't require scripts or programming knowledge, right? So although we require R for some of the plugins within SeqGeek, which we'll talk about in a little bit, overall, it doesn't require you to enter any kind of third-party software programming. So everything you need to get started with your analysis is available inside of SeqGeek, and you don't have to execute any programming to get visuals or get data. And if you're familiar with FlowJo already, which is our flow cytometry analysis platform, you'll see that the point and click interface is going to be very familiar, right? So if you already know how to use FlowJo, or if you want to just watch 1 or 2 intro to FlowJo webinars, you could learn a lot about just navigating the software, because you'll find that a lot of the buttons and a lot of the functionality end up being in the same places, right? So very visual friendly, very interactive and approachable compared to having to run programs and scripts in another software. And you can immediately explore your own data, right? As I mentioned before, sometimes the holdup with our analysis is that we need to identify a collaborator not only willing to help us with the analysis, but who also has the time to really produce some meaningful results in time for some kind of a publication, or if you're trying to present this as an abstract, or you just want to get some preliminary information from your experiment. And so you can really have the power to just take your files from your instrument, from your experiment, and immediately start to explore them in the software. It's compatible with various single-cell acquisition platform matrices, so it's not written for a particular instrument in mind. but it is compatible with other platforms, including like 10x Genomics, for instance, or the BD Rhapsody platform. Also, the software also has multi-omic capabilities, right? So if you've used the Rhapsody and done some kind of AB-seq experiment where you're looking at protein as well as mRNA transcripts at the same time, you can handle that kind of analysis within SeqGeek as well. So it's not limited to just single-cell RNA-seq in terms of transcript analysis, but you can also get something similar to flow cytometry information within the same platform for analysis. So here's an outline of what we're planning to cover in today's webinar. So I'm going to start by just giving a quick orientation of the SeqGeek workspace. I'm talking about how to get around in the software and what the graph windows mean. And then we're going to just go right into a kind of introductory workflow on how you can approach, at least as a starting point, a single-cell RNA sequencing experiment that you are analyzing within SeqGeek, right? So we'll talk about normalization and quality control, gene sets and enriching for what those might be, dimensionality reduction and clustering, which are some computational tools to kind of do a more automated approach to your analysis. We'll also get to the heart of differential expressed genes, so how to ultimately form these kind of transcript profiles between your samples or between populations in your samples. And lastly, I'll touch a little bit on plugins and pipelines that are available on the FlowJo exchange and how you can incorporate them into your analysis. I won't show a lot of them today because some of them take a couple minutes to run and are a little outside of the scope of just a basic introductory workflow, but I'll touch on where you can find them and what they can do for you. All right, so first let's talk a little bit about just navigating the SeqGeek software. Okay, so when you launch SeqGeek, this is really where you're going to spend most of your time. So this is called the SeqGeek Workspace. Okay, so this is the interface to load and analyze your samples. So it comes in 3 main sections. So your first section is going to be the activity ribbon, organized by tabs. So each of the tabs are going to have different functionality in the form of buttons where you can execute all of the different functions of the software, including some of the things I've just mentioned, like dimensionality reduction, clustering, quality control. So much like some kind of a Microsoft application where you have all of your buttons toward the top, that's basically how your workspace operates as well. Okay, so if you need to execute something, it's likely going to be somewhere up top. And next we have our gene sets. So we'll talk a little bit about what those are, but just know that when they're formed, they're going to show up here. And so you'll see your gene sets indicated by these different symbols. And the count is just going to be how many genes are in that specific gene set. All right, and then below that, we have our sample pane, which is going to house all of our samples as well as the analysis for our samples. So you can see the list of the files that you've loaded into your SeqGeek workspace. And then in the pane below, if you're doing any kind of population formation with gates, you'll start to see that hierarchy or that ancestry form down here in this pane. The statistic beside it is just going to be the frequency of that population from the parent, right? So in this example, we have CD8 positive cells that are 50% of the CD3 positive cells. Okay, so percent relative to the ancestor population, right? And then you'll have number of cells or events. That's going to be the last column. And these columns are customizable. You can add columns if you would like to add some additional information to your workspace. But this is essentially what you'll be working in and how you'll be organizing your analysis in the SeqGeek software. Again, if you've ever used FlowJo before or even just opened it, you'll see that it looks very much like FlowJo, with the exception of the gene set pane and also just the color scheme. All right, and then again, much like FlowJo, we have graph windows, right? So this is where you're going to interact with your sample so that you can actually visualize your events or your genes in the form of dots. And this is ultimately where we're going to draw gates to form either populations or gene sets. So one of the key features of SeqGeek and its difference from FlowJo is what we refer to as the pivot window, right? So when we're doing single-cell RNA sequencing, although we're doing that experiment on cells, and it's going to be important to establish a cell view, where I can view each individual cell, I'm also interested in transcripts, right? So I'm going to also want to see individual genes and where they fall against my cells. And so that's really the power of the cell view and the gene view, because using these two in combination, you can start to ask these questions about which cells are expressing which transcripts. So for instance, if we're in the cell view here on the left, this is kind of a traditional view you would see in, say, a flow cytometry analysis or in FlowJo, whereby each dot on this plot represents a cell or an event. Okay, so we have the x-axis for some kind of a surface parameter like CD4 or CD8, and where that dot falls denotes kind of the profile of that cell, right? So we have a dot in cell view, which means that this cell is positive for CD4 and positive for CD8. Or as in the form of this gate, we have an event or a cell that's positive for CD4, but negative for CD8. And when I form gates of interest, like the two populations I just pointed out, then those populations are going to form a hierarchy in our sample pane, like we described on the previous slide of the workspace. Okay? So in cell view, each dot is a cell or event. And if I make a gate on those cells, I'm making a population. That population hierarchy will show up in my workspace. Okay? So kind of a traditional approach to flow cytometry. Now, if we pivot to gene view, which is just the click of a button, we can see that the view changes a bit, right? So now each parameter on my X and Y axis is a population, right? Either a population I've found myself, or it could just be your whole sample, right? So here you're saying B cells and T cells, and each of these dots now represents a gene or a transcript, right? And you can't tell here because this is a static image, but if I were to hover in the software over one of these dots, you would see that gene name appear, okay? So populations on the axes, and then each of the dots now represents a gene. And if I make a gate on genes, right, you're not necessarily making a population, right, because these aren't cells, these are genes. But if I gate on those genes, or a group of genes, I will form what's called a gene set, right? And so that gene set's going to show up in the top pane of my workspace, right, that we just talked about on the previous slide. And then what can I do with these gene sets? Well, they can be inspected or opened, and you can see which genes fell into that gate. Okay, so a gate is essentially kind of a net or a filter of something that you're trying to capture. So in the case of cell view, you might be capturing a certain population of cells, but in the case of gene view, you want to capture a group of genes for some particular reason. So in this case, I have B cells on one axis, T cells on another. And I can see that the genes that are in the orange gate are kind of enriched in my B cells. So maybe I want to know what those are, draw a gate around it, create a gene set, and ask, what are these genes that are enriched in my B cells, right? And then I can get that list here and see what those genes are. And I can do the opposite, right? I can say, well, what genes characterize this population that's on the y-axis? So then I can make a gate there. Similarly, make a gene set, open it up, ask what these genes are that make this population unique from the one that's next to it. Okay, so this is really going to be the power of the SeqGeek workspace when we're going into our analysis, is not only the graph window where you can, change colors and make gates, but the power of the pivot view where you can inspect cells individually, but also get the transcript information as well. All right, so we talked about gene sets. Well, what can I do with those, right? I already mentioned that I can open them up and see what genes are there, but it doesn't have to end there, right? You can get some more information from that. So again, gene sets are going to be a way to organize your genes of interest into manageable groups, right? So we talked about an analytic gene set, which you would form from a gate when you're in gene view. So once I've made this gate around these genes that seem to characterize my B cells, this gate in orange, can draw that gate and ask, well, what are those genes? And inspect them manually myself, get some statistics as like, what's the fold change of upregulation? What's the p-value between the two different populations? And then ultimately you can take this gene set, export it, and do some kind of a gene set enrichment, right? To ask, well, what are these genes? What kind of pathway are they involved in? What kind of function do they have, right? And so this can be done natively within SeqGeek if you import gene sets, some of which are included with a free trial download of SeqGeek. Oops, let's go back one. So you can import gene sets and do that one way and do an enrichment test looking at gene sets that you've imported. But another way to do it is to use web tools, right? So I can export this gene set, turn it into an Excel file, pop that Excel file into a web tool. Just one example is Panther, and ask that web tool to tell me where do these genes fall in a pathway analysis? What kind of pathway are they involved in? And then I can learn about my population here that I've zoned in on for those transcripts and learn what kind of pathways have been upregulated or downregulated, and really start to make comparisons between populations or even between samples. But in addition to an analytic gene set, you can also have a static gene set, right? So this is something you would create manually from a list of genes or just from something you've imported, like I mentioned down here. So why would you want to create a manual list of genes? Well, maybe there's a group of genes you're already interested in, right? Maybe this is a new drug you're testing and you know it's going to activate proliferation of T cells, right? So you want to look at T cell lineage genes, and you can create that list by just giving it a name, searching for your genes of interest, adding them here, and now you have your own static gene set that you can visualize within SeqGeek or ask if it's enriched in any particular populations, right? Maybe you have another pathway of interest like mitochondrial genes, which could be associated with cell death at times, or apoptotic genes. So you can make your own lists as well, and that you can work with them within your analysis. So you don't just have to do these analytic ones through gates, you can also create your own. All right, so we've talked about some of the features that kind of make SeqGeek and why you would have those tools and what they can do for you when you're in this analysis. Okay? So now that we've talked about those features, let's go ahead and just go into a basic workflow, right? So once you're in SeqGeek, what do I do with that information and how do I navigate through the software? Okay, so we'll start with talking about normalization and quality control, just some pre-processing steps to do before your analysis. And although there's a lot that can be done manually within SeqGeek, you can certainly make your own gates and just kind of nose around and cherry pick populations of interest. We're going to talk about the power of computational tools like dimensionality reduction and clustering, which are really going to save you a lot of time and give you more of a discovery-based approach, right? So we're going to talk about, after doing our pre-processing, moving into some algorithms where we can kind of get things moving and start to visualize some of our data. And then lastly, ultimately, we want differential gene expression, right? I want to know what makes a population different from another, what genes are upregulated in one population over another. So this is ultimately going to be the heart of the analysis. And I think it's worth saying that although I'm showing this in kind of a linear view, keep in mind that particularly for the last 3 steps, you know, they can really be done in any order. And if at some point you decide you don't want to do dimensionality reduction, you want to go right on to clustering or vice versa, you can certainly do these in an order that makes more sense to you. This is just the order I'll be presenting it in. Okay? All right, so let's talk about normalization and quality control. So this is going to be some pre-processing for your downstream analysis. Oh, keep going ahead to slides here. Let's go back one more. There we go. So let's talk about normalization and quality control. So normalization is going to be an adjustment of your data to a common scale. Okay, so why would we want to do that? So across your samples, you might have different numbers of reads, right? So maybe in one sample you have 1,000 total reads, and maybe in another sample you have 10,000 total reads, right? You're able to sequence more there. So this is just going to put them all in the same playing field, right? So it's going to normalize that scale across all of the samples by saying something like, oh, we're going to set it all to how many counts per 10,000 counts total, okay? So it just puts them all kind of on a level playing field. And so all it'll do is just adjust the scaling. So if this is the raw information, you can see all that data has kind of just shifted over to the right to a more larger scale rather than being kind of crunched here on the axis and not really capturing the range of the information because some samples might be getting cut off. Okay. So it's just going to put everything on kind of an even scale. And the other benefit is that, particularly if you're doing multi-omics analysis, your range and your scale for your proteins is likely going to be much larger than for your transcript reads, right? So those might be in the dozens or the hundreds or the thousands, as where your proteins are likely going to be, you know, more on a log scale in the thousands or 10,000s range. So by doing a normalization like this of like counts per 10,000, you have a better chance of kind of keeping everything on the same axes, and you don't have to keep going back and forth to capture data that's going off scale. Okay. So that's normalization. So what about quality control? Okay, so this is going to be ultimately just a way to clean up your data, right? We want to remove events that are kind of ambiguous or questionable, right? Like dead cells and doublets and things of that sort. So this is an automated platform within SeqGeek in the sense that it's the launch of a button, and it already launches these 3 graph windows, and it already sets up the axes for you. All you have to do is the gate placement. So you as a scientist decide where you want to place these gates, but we can provide a little bit of guidance. Okay? So the first quality control filter is just going to be to capture quality cells, right? We want to remove outlier events, which could be empty wells or doublets. So within cell view, you know, we want to take cells that all have kind of follow a similar ratio for library size and genes expressed. As we go further out of range here, you know, you might end up with wells that had too many cells or cells stuck together. And if you go too low, maybe those ended up being kind of empty, right? It's just noise. So we want to capture kind of the bulk of the cells, removing outliers. So this is going to be our first quality cells filter. The next 2 are going to be done in gene view off of the quality cells. So we're going to pivot to gene view and then do some quality control on our transcripts, right? So the first one's going to be what we can call an outlier gene filter. So here we're trying to remove genes that are expressed by few or all cells, right? So for an RNA sequencing analysis, you're going to get a lot of housekeeping gene expression, which isn't going to be very likely informative for your analysis. So you're probably going to want to get rid of those kinds of genes, which are going to be expressed by all your cells. And the converse is you don't necessarily want transcripts that are expressed by only a few cells, because it's probably not really been some kind of a global change across your sample, but rather it's just you know, 1 or 2 or 3 cells that are just had a kind of an abnormal reaction to some kind of stimulus or some kind of disease state. So we want to get rid of kind of that background noise and also get rid of genes that are just kind of widely expressed, because then they're just adding noise to the system. So we want to go right in the middle of this sweet spot where we have genes that are kind of expressed by most of the cells, but not too few and not all of them. Okay, so that's our first gene filter. And then we're going to want our highly dispersed genes, right? So off of our outlier genes, so right in this sweet spot, where we want the genes that are, you know, kind of expressed between most of the cells, take those genes and then ask which ones are highly dispersed. In other words, which ones show a lot of kind of dynamic range of expression across the cells, right? That are a lot upregulated or a lot downregulated, right? Because that's what we're ultimately looking for, is changes that are going to help us define subsets of populations within our sample. Okay, so we're going to capture the ones that have high dispersion or basically high variability. Okay. So this is just some pre-processing before we move into our analysis. All right, so when we go into our analysis, you know, as I mentioned, we can certainly do lots of things manually, right? We can start looking for cell types of interest based on their transcript profile, or if we have an AbSeq experiment based on, you know, what they're expressing on the surface of the cell, maybe I'm looking at only CD4 T cells or only CD8 T cells, something like that. But it's not entirely an unbiased view for approaching your analysis, right? And it also can be quite laborious depending on, you know, how many different populations you expect to be in your sample. So computational analysis is really going to be a great tool for data sets like single-cell RNA-seq, because they can help you tackle the complexity of your data using a more discovery-based approach. That's ultimately going to also save you a lot of time, but help you uncover possibly rare or novel populations in your sample. Okay. So just as a way to illustrate, the value of computational analysis and kind of just really strip it down to bare bones, what is it and what does it do for you? Let's walk through this experiment. Let's say I gave somebody 4 jars of jelly beans, right? And each jar has about, let's say 500 jelly beans in it. And there's maybe 20 different flavors in each of these jars, but I want them to identify the jar that has the most of my favorite flavor, which is the bubblegum flavor. Okay, so you're gonna have to go through each of these jars, sort them all out, divvy them all up, and count, the frequency of that bubblegum jelly bean, and tell me which one had the most. Okay? And also there might be a mystery flavor in there. And I want to know how many are of that one as well. Okay, so you can see how it kind of sounds like a mouthful to have to go through all of this, especially if you had to do it by hand. But with something like computational analysis, we can automate that process, kind of, dump all of those jelly beans together on the table, arrange them by size, by color, by flavor, and also, segregate them into these, let's call them wells, and then be able to kind of more easily digest, which one has more of the bubblegum flavor if we divide them into 4 different trays with all of these different wells. Okay? So this is really where the power of dimensionality reduction and clustering can be seen, right? So with dimensionality reduction, we can kind of lay them all out nice and flat so we can see them and have them kind of sorted by different colors or different flavors. As the clustering is going to be kind of these wells, right? So this is going to be a hard separation between all of these jelly beans so we can start to inspect them and classify them and ask what they are. Okay? This is kind of an oversimplified view of what these tools can do for you, but it's just an example of what would be the end product from something like a large single cell RNA-seq experiment where you have thousands of cells with maybe hundreds of different transcripts, and how you can get to an endpoint of something like this that's a little bit more digestible. Okay, so let's start with dimensionality reduction. So again, much like the jelly bean experiment, right? It's just a way of kind of throwing all your data on the table and flattening it out so you can see it all at the same time and kind of organizing that data where it kind of like ends up with like, right? So you're compressing your entire data set into a 2-dimensional space, right? And so that can be seen as an example down here. So these are t-SNE plots, or a t-distributed stochastic neighbor embedding. So this is just going to be a low-dimensional representation of your high-dimensional data. And so as an example, you can see that these are the events from all of the events in an experiment, but similar events have kind of been grouped together in these islands, right? So all of my events that have CD3 transcripts have all ended up in this island. The ones that have CD14, the ones that have more of it have kind of ended up on this island, less of it maybe on this island, right? CD19, they've ended up in this island. So you can start to see how, like ends up with like when you do a dimensionality reduction, and it allows you to see all of your data all at once, so you don't have to have various plot windows just to see each of these individual markers. So within SeqGeek, there's a couple of options for dimensionality reduction. So there's t-SNE, like I just mentioned, but we also have PCA, or principal component analysis, as well as LDA, linear discriminant analysis, right? Those can be launched right from within FlowJo. So we have our dimensionality reduction platform, which when opened will prompt you with one of these choices. Okay? And ultimately, the end product will look something like this, where you have your low dimensional embedding of your high dimensional data on your bivariate plot. Okay. Now, there are some other dimensionality reductions, but in the form of plugins, right? So these are plugins you would have to install from our website that require our installation as well. But once set up in SeqGeek, you can execute these and have some other options for dimensionality reduction, including UMAP, which is kind of growing in popularity in the field, as well as TriMap. Both of these are kind of favored for more trajectory studies. You get a little bit more information in terms of global structure, being able to interpret cells that might be in transition between islands a little more than you might with t-SNE, but t-SNE does flatten very well, so it's also kind of a crowd favorite. All right, so clustering. So if dimensionality reduction is just laying out all of our events clustering would be, forming gates around those events. Okay, right. So unsupervised identification of populations that are ultimately going to become populations in your workspace that will be automatically created for you. Okay. So this can kind of be visualized here, where if you had all your events in kind of a high dimensional space, and they would all start being separated by similarity in terms of their transcript profile, or if you have a multi-omic experiment in terms of their protein profiles. And then, we're going to code them into different populations and make those subpopulations. And it basically just give them numerical names because the software is not going to know what these populations are biologically, but it does know that for some reason or another, they're very similar, and it's placed them in the same cluster. Right? So when you run your clustering algorithm, it's essentially going to give something like this. So it's just going to create, let's say in this instance, 19 different subpopulations or 19 different clusters. Okay, then it's up to you as the researcher to look into these clusters and figure out what makes them unique and which ones might be of interest to you because they're of a higher frequency in one sample or another. Okay. So the kind of the power of clustering can be illustrated here in this publication here that describes Phenograph, which is one of our clustering options in the form of a plugin. Right, so here they've done a dimensionality reduction of a t-SNE, right? So laid out all the data points so we can see it all up front in a bivariate plot. And they've taken their data and run it through Phenograph, right? And asked it to find populations completely unsupervised. And the software identified 15 different populations, right? Color coded here on this low dimensional representation or on this t-SNE space. But if the authors take that same data and just manually through their own knowledge, gate populations in that sample they know exist, you can see that they find roughly the same number, and they can find populations that correspond to an unsupervised identified cluster down here, right? So they can find this population manually, which they identify as a mature B cell just from their own immune knowledge and from the marker profiles. But the Phenograph algorithm has also found what it thinks is B cells, but it doesn't know it's B cells because it doesn't have that biology information. But it found a population that was very similar to one another. The events that are inside of it created its own cluster. Okay? And so I can say that it's likely that the clustering algorithm, found all of these populations much faster than a user would be able to do if they did this all manually. Okay? So that's kind of the power of clustering in a nutshell, right? So you can find populations that you would otherwise probably be able to find manually unless they're really rare, but you can automate that process and then visualize it on a t-SNE space. Okay? So in terms of clustering within SeqGeek, so there are some kind of native options, right? We have k-means, k-nearest neighbors. So k-means is going to need a little supervision, asking you to kind of guess how many clusters you think would be in your sample. And k-nearest neighbors is going to need a training data set. So this is why folks have started kind of favoring plugins that offer other clustering options like Phenograph, because it doesn't really require a training data set or for you to estimate how many clusters you think need to come out of the data. So it's a little bit more approachable if you don't have a whole lot of knowledge about what you expect to find out of your sample. All right, and then getting to differential expression, this is ultimately what the endpoint of your analysis is going to want to be, right? You're going to want to elucidate transcription profiles between different populations in your sample, right? So let's say you've run some kind of clustering algorithm here, and now you've identified cluster 4 is very interesting, right? I think cluster 4 is got some unique transcripts in it, and I want to see what's in there. So you can just use some Boolean gating and make a, something that's up in cluster 4 relative to the rest of your clusters, right? Open that up in gene view, right? And now here we have a volcano plot showing the upregulation of genes within cluster 4 and the downregulation of genes within cluster 4, right? And we make a gate, right? And when we make a gate in gene view, we create a gene set, right? So if I look at that gene set and ask, what is upregulated in cluster 4, I can see that, right? So I can see that I have some genes that get upregulated, And if I do some kind of a gene set enrichment with either imported libraries or using something like Panther, like I described before, then I can see what those genes are, right? I can see that there's genes that are enriched for monocytes. There's genes that are enriched for B cells, right? So you can see that most of them are falling up in the first two. So you can start to figure out what's inside of these gene sets within each of your clusters and ultimately identify what those genes are and what kind of pathways they're involved in. So this can all be achieved again manually. You could do your clustering. You could compare populations on the X and Y axis, launch the differential expression to get these volcano plots, and then make gates, make gene sets, export those gene sets, and ask what they are. But you can automate a lot of the first part of that process using plugins, right? And I'll talk a little bit about these in the demo. So we have one called ICLR, which can help you automatically create these gene sets and tell you, what makes your clusters unique from the rest. And then we also have Seurat, which is can also do this, but it's also a full pipeline, and it's going to do a lot of other things as well, including quality control, dimensionality reduction, clustering, as well as this differential gene expression as well. Yeah. All right, and so you've done all of your analysis, right? So now you need some kind of visualizations that you're going to put in your publication, or you're going to put in your notebook, or some kind of a presentation. So SeqGeek really has you covered on that front as well. You don't have to export just a bunch of numbers into a third-party software, right? You can write within SeqGeek, take your own data, and start to create the visuals that you need to send your message across. So within the layout editor, we can make reports for each of our samples with just showing some bivariate plots or t-SNE plots. But you can also create heat maps. So if you have genes of interest that you want to highlight between different clusters or between different populations, you can select those genes and have them displayed to you in a heat map view. You can also do overlays, that's an example down here. So maybe an overlaid histogram to show the variable expression of a gene across your different clusters. Or you can do a t-SNE overlay. So if you've used manual gating, or if you've used a clustering algorithm to identify populations of interest, you can go ahead and overlay that onto a t-SNE, and we can see it all at the same time, much like we saw on previous slides for the phenograph algorithm. If you had multiple samples, you could make one graph for each sample and see if you have populations that are missing. And everything is very customizable, right? We can change the contours of our lines, the type of dots we have, whether they're open or whether they're closed. We can also change colors, right? So again, you're making all of your figures within the SeqGeek platform. And all of these figures can be exported as images or as a different type of file format, like a PDF. There's also an ability to get statistics. So if you do need hard numbers, that can be exported from the Workspace as a CSV file. And so there's a lot of native functionality for formatting your figures, but there are also some plugin options. So violin box is kind of demonstrated here on the far right. So this is going to make violin plots or box and whisker plots if you prefer. And basically, you can load up 2 genes of interest across 2 populations, either that you made manually or through clustering, and ask, what's the expression of that gene between the 2 populations. You can even generate some statistics, and you can show each of your events as dots over your violin plots if you'd like. And the images are downloadable. You can put them in a PowerPoint or put them in your lab notebook. And there's much more on the FlowJo Exchange, right? So I've done a couple plugin shout outs throughout the talk and talked about where you can incorporate them into your analysis if you want to venture outside of the SeqGeek software, bring them back in and be able to use them there. So these are just a couple of shout outs for some of the plugins that are valuable to a SeqGeek analysis. So for quality control, we do have a bachelor plugin, which is going to be for batch correction. Right, so we don't want islands to be completely attributed to one sample or another, particularly if they're just replicates or different healthy donors or something like that. Right, so we want to make sure that there's real biological variability and not just batch effects. So we do have a batch correction algorithm for that purpose. And we also have Euclid's quality control your clusters to make sure that they're uniform, right? We want clusters to be very different from one another, but we want events within a cluster to be very similar to one another. Okay, so this is going to help you check that. Dimensionality reduction, right? We talked about the options that are within SeqGeek, but there are some other options, UMAP and TriMap in the form of plugins. Clustering, we have phenograph. If you don't want to use the k-means or k-nearest neighbor options within SeqGeek. Visualization, we talked about violin box already, so how you can maybe show some gene expression across your populations. But we also have some population hierarchy plugins. So if you've formed a population hierarchy in your sample pane and you want to show kind of that gating strategy or that ancestry, these make kind of like tree plots to show kind of the ancestry of your populations and how you got to, say, activated CD8 cells from your bulk population. And then we also have more discovery tools and full pipelines, right? So Seurat is going to be one of our full pipeline tools. And so here you can see that it really does a lot. So it's a multifaceted tool. You can do quality control if you don't want to do that within SeqGeek, normalize, dimensionality reduction, clustering, and it's going to give you some visualization tools as well. So kind of automate that process of making all these t-SNE overlays, and it's going to help you out with that automatically. ISLR can do some 3D visualization for your dimensionally reduced space, but it can also do differential gene expression and kind of automate finding genes that are up or down in particular clusters. Monocle is going to be pseudo time, so if you're interested in cell trajectory, and VDJ Explorer, TCR, BCR recombination, if you are interested in kind of profiling those repertoires in your samples. All right, so there's going to be a lot of stuff out there that you can use within SeqGeek, a lot of endless tools, a lot of endless workflows, but we're just not going to be able to get to a lot of it today. But I do want to leave you with, the knowledge that there's a lot more learning that can be done on our website. And I just want to point out those tools, right? So we have webinars like the ones you're attending today, which are hosted live monthly. So you can bring your questions to us, but they're also recorded. So you can access our library of recordings. We have FlowJo University. So these are going to be 5 to 15-minute video tutorials, really just point and click functionality, not a whole lot of deep dives into topics, but just a quick way to learn a new method in SeqGeek. We also have a searchable web manual, right? So it's not just this giant PDF you have to download and read through. It functions more like a website, so completely searchable by topic. And also you can download from our website a free 60-day trial. So quite generous trial for 60 days. And within that trial, you can also get access to some demo data. And if you're ever having any trouble with the software and you've already installed it, but maybe you can't get plugins to work or you're having trouble seeing your data on SeqGeek, feel free to contact myself or tech support, SeqGeek@bd.com. And we'll be able to help you out and get you going in SeqGeek. All right, so that's all I have for slides. I realize I went a little over the 30 minutes, but that's okay. I'll try to move a little more quickly through the demo. Before I do that, I'm going to just take a quick field trip over to the FlowJo website to show you some of these resources. Right, so we talked about all of the learning materials. So at flowjo.com, if you head over to the Learn tab, you can see our webinars, FlowJo University, and SeqGeek Software Help. This is going to be the searchable web manual. I'm just checking a question in the chat. Let's see. Oh, that is a great question. The question is if there are any publications that are comparing this platform against others. To my knowledge, I'm not aware of one, but I will say a lot of the tools that we gather into SeqGeek are published themselves. We've kind of just curated them into the SeqGeek platform. So if you wanted to learn more about the algorithms and which algorithms to choose within SeqGeek, you could probably find external publications for that process. That's a good question. All right, so there's all the learning materials. So if we go over to downloads, gonna work. Here we go. Here we go. And so here's SeqGeek. You can download it and it can be a trial which you can activate with your email, and you can have that for 60 days. And during the 60-day trial, really take advantage of that time. And if you don't have data of your own, there are some data sets that come included in the downloaded package once you install it. And you can access some of that demo data. And actually the data set I'll be using today is part of the demo package. So you can access that same exact file if there's anything you want to repeat on your own time. Okay, so now we are in SeqGeek, right? So this is the workspace. So let's orient ourselves again, right? We have the activity ribbon. We have the sample pane where our samples and analysis are going to form. And then up here, we'll have our gene sets once we start forming those. Okay. So before I load any samples, let's go ahead and talk about the preferences button as well. So if you hit that button, preferences, right? So this is where you're going to be able to, get some information about the software, set it up, and also be able to customize your experience. Okay. So I'm not going to talk about it a whole lot today, but once you download R and install it, and once you download plugins and install those, you'll need to make sure to tell SeqGeek where the information is, and this is where you'll do that. So in your preferences, diagnostics, you will set your R path. So tell SeqGeek where to find R, and then tell SeqGeek where to find your plugins. And so we have plugin installation videos on our website, so I'd encourage you to watch those to get the details. But this is where you'll come to set those up. If you want to customize fonts, make them bigger or smaller in your workspace, this is where you can do that. You can also customize gates, right? So if you want your gates to be a different color, maybe you want a bright pink color so they pop out more on your screen, or you want a nice thick line instead of a thinner one, you can customize that experience as well. So just know you're not always stuck with the defaults in the software. You can come in, see if there's a way you can customize that experience. Okay, so loading samples. So a couple ways you can load them. You can always hit the add samples button. So remember, a lot of our functionality is going to be towards the top. You can hit the add samples button, which is just going to open a browser, ask you to find your samples. But SeqGeek is also very drag and drop friendly. So you can feel free to just drag your samples right in here, which is what I'm going to do. So I'm going to be using the 6K PBMC data set. So this is just human PBMC from a 10x platform. So I'm going to go ahead and just drag that right in. It's going to take a second to read it. It's a pretty large data set, so it needs a minute just to get going. But I can see that it's working on it. There it is. All right, so we can see that here's my samples. Here's my 6K PBMC data set. It's also showing up here in my sample pane. Okay. So first thing we're going to want to do is probably do some normalization, right? So we're going to want all of our reads to be on the same scale. So we're going to go ahead and hit that sample and launch the normalization platform. So I'm going to hit that. And pretty simple, it's just going to ask me to pick the sample. It's going to ask me which genes I want to normalize. Right, so if you're doing a multi-omics experiment, you might want to make sure you only pick your genes and not your proteins, but this is only RNA, so we're not going to worry about that today. So we've gone ahead, I'm going to do all of my genes, and then how do I want to normalize it, right? Do I want to do counts per 10,000, counts per million, or customize it? Popular in the field to just pick the 10,000, so we're going to normalize counts per 10,000. Right, we're going to hit run. And it's done. So how do I know it actually worked? This little symbol next to your sample, right? The symbol matches the same symbol that's in the normalization button. It's going to turn a blue color, right? So earlier it was gray. And once I did the normalization, it turned this blue color, right? So that's how I know the normalization actually applied. Okay. Right, so now we've normalized. So now we want to do some quality control, right? We want to get rid of dead cells. We want to get rid of housekeeping genes, and we want only, highly variably expressed genes. Okay, so let's pick that sample again, and let's go ahead and launch the quality control platform, right? So hit that button, and it's going to launch automatically 3 graph windows. Okay, so let's take these one at a time. So let's make this first one bigger. Okay, notice how we're in cell view. Okay, right? So each dot is going to represent one cell, right? And I'm looking at library size over gene expressed. And I'm going to want to pick the, the area where most of my cells are falling, so they're kind of following the same distribution. And we don't really want these outlier cells that might represent doublets, and the kind of lower end cells that might represent empty wells. Okay, so we want to capture kind of this dense area here. So one way I like to look at this is in the log axis, and really that's just because I want to be able to see kind of the lower end events a little bit better. So how do we change our axes, right? How do we manipulate the white space? There's a T button in the graph window beside each axis here, T for transform. And if I hit that button, I get a few kind of hot link options here. So earlier I was in linear axis, I picked log. But if I wanted to customize that, I could just go to customize axis, and I'd get some nods to help kind of eliminate white space if I wanted to do that. Okay. I'm going to go ahead and keep the default, right? So how do I capture these events? Well, we're going to do that with a gate, right? So a couple of different gating shapes and options here. I think in this case, a free-form polygon might be best, right? But there's also a rectangle, also a circle. We also have an auto gate function, which I'll show that one. It's just going to automatically, depending where I place my mouse, going to try and capture the densest area of where my cells are, right? So maybe that's a good place. And maybe I'm going to call this, you can of course call it whatever you like. I'm just going to call it quality cells, say okay. Once I've dropped it, I still can manipulate the space a little bit if I want to, right? And if I go down to my active gate, I can make the gate, a little bigger, can make it a little smaller if I want to, without having to manipulate each of these little vertices. You could also change the color of the gate, if you decide you want to give it a different color, right? So I've decided I want my QC gates to be red instead of black. I can do that. Okay. You can also change the view if you're unhappy with pseudocolor. So pseudocolor is going to be kind of a heat map for the distribution of your events, right? So where the events are falling the most, or where you have more cells falling, it's going to be red. It's going to turn blue as you get further out into the outlier events. But if you want just a traditional dot plot, you can change that down here in your options. You could do a contour. Maybe you still want to see your outlier events, you can check that box, right? So a lot of different ways that you can just customize your view. I tend to favor pseudocolor, so I'm just going to stick with that. But just know that you can change colors down here, as well as the color of your gate. All right, so in cell view, for quality control, I've made a quality cells gate. So since I've done this in cell view, that population, we can see it in my sample pane, right? And notice how it's offset from my sample, because this is an offset of the ancestor population, which is basically all my cells, right? So all my cells, from that, I did a quality cells gate. Okay. So there's our first quality control. So for our next quality control, right, we're going to be pivoting to gene view, right? So now I want to capture genes that are expressed by most of my cells, but not too few, and definitely not all of them, because those might just be housekeeping genes. Okay. So we're in gene view. We can tell that this is white instead of black. So here, another case where I might want to adjust the scale. So go ahead and change that to a log axis, just so I can see it a little bit better. Right, and so now we can see kind of this plot that we looked at in our talk earlier today, right? We can see that these are probably going to be some housekeeping genes. These are probably just going to be, rare transcripts that are only expressed by a couple cells here and there. So we want some kind of a sweet spot in the middle. So in this case, I think I'm going to use a rectangle gate. Go ahead and, maybe I'll place it somewhere there. And I'll call this outlier gene filter. All right, maybe I've decided on that's being a little strict. Maybe I'll go ahead and do that. Okay, so here's my outlier gene filter that we've done on gene view. So notice when I made this gate that it formed here in my gene sets, right? Because this is in gene view, this isn't in cell view, right? So this is a gene set that I've created. Okay, so now the last quality control step we'll do, see if I can get this window to move. There we go. All right, let's make this a little bit bigger. All right, so last quality control step, right? We want the highly variable genes, right, that are variable in expression. But I don't want to do it on all my genes. I want to use this filter I made and kind of transfer it to this next window, right? So off of my outlier genes, I now want my highly dispersed genes. So we're going to hit the all genes button and let's pick only the genes that pass this filter, right? Let's hit OK, right? And you can see I have less dots now because I'm only capturing the ones in this gate. If I'm having trouble seeing, right, it doesn't look like I have a whole lot of genes up here, or definitely not as many as down here. If you go over to your display, right, you can use large dots if you want, help yourself out a little bit. Again, I'm going to go with a rectangle gate, maybe capture like just right off that plateau there, right? And I'll call these highly dispersed genes. Oops, I didn't mean to move my gate. I was just trying to move my population name. There we go. Okay, so I moved my population name. So we can see, right, how our filtering is going in place, right? We started with, what, about 20,000 genes, went down to 8,000 after the outlier gene filter, and then went to 172. Now, if you feel like you're being a little too strict, like you think you're losing a lot of information, you know, there's no reason why you can't be a little more generous with these filters, right? It's completely up to the user. And that's why it's nice to do the quality control yourself rather than leave it to kind of an automated algorithm, because then you can kind of see, you know, what's being kicked out and if you feel like you're kicking out too much, right? All right, so we've done our quality control. So one thing I like to do before I close this platform is to just go ahead and copy these plots to the layout editor because they might not be as easy to get to once you start your analysis, right? So the layout editor is going to be this L ruler button here, right? This is basically your blank canvas for kind of making your reports, right? So if I come here, quick shortcut is to copy to layout editor, right? So if I hit that, there's my first plot, right? There's my quality cell plot. If I go to my next one, I could just hit on a PC, Control L, right? That has also made its way now to my layout editor. And same thing here. Right, now I've got all of my quality control plots here in my layout editor, ready to print or export. If I decide to, I can even rename this. Instead of calling it layout, let's call it QC, right? And then I can find it quickly. If my PI asks, well, how did you quality control this data? You've got your layout editor ready to load and show those plots. Okay? Okay, so let's go ahead and close these. So another quick shortcut, close these all at once. File, close all graph windows. It saves me a couple clicks there. All right, and let's open up my workspace again. Okay, so we did our normalization, right? We did our quality control. So let's go ahead and move forward with a dimensionality reduction, right? So now we're going to go ahead and compress all of our data into a dimensionally reduced space, so we can start doing some visualizations. Okay? So we're going to launch the dimensionality reduction platform, right? Go ahead and open this dialog window. So if you're interested in using something like a t-SNE, it's often best to go ahead and compress the data with principal components first. And really that way you do kind of a pre-digest before you major digest, right? So t-SNE will handle smaller pieces better than it will just these massive amount of transcript data. Okay? So if you're interested in doing a t-SNE, go ahead and run a PCA first, right? So we're going to do a PCA, which is still a dimensionality reduction, but this is just going to be our first pass. PCA, you can name the run if you want to. Do we want to do all the genes? Well, probably not, right? Maybe we want to just do the one that passed our high dispersion filter, right? All the ones that are going to give us the most information about variability in our populations, right? So I'm asking this graph window here to just pick my highly dispersed genes, going to add those, right? Which is only 200 instead of what, 20,000. Right, so I'm going to select that, select those genes, principal components. I'm going to just let it run the default 25, but if you want to go ahead and dictate how many it runs now, you can do that. So I'm going to go ahead and run. All right, now here's our principal components, right? Let me move my Zoom controls over for a second. There we go. All right, so here's my principal components, right? So I calculated 25, but you may not need all 25, right? So it kind of becomes up to the user. So, how many principal components do you want to take into your analysis? Well, you might want to just take the ones that follow one of two rules, right? So some people like to follow the 1% rule, right? So once my variability goes below 1%, maybe I don't want those principal components, right? And then some are the school of thought that you typically don't need more than 15, right? 15 principal components is perfectly fine for your data. You don't need to take any more with you, okay? So it's up to the user. You can try different approaches, see what gives you the best results. I think in this case, I'm going to just go ahead and go with the first, let's just say the first 15. And then I'm going to tell SeqGeek to just give me 15 PCAs back, right? I don't want all 25 after all, right? So it's going to give me back those PCAs, the 15 that I asked for, right? And so those are just going to show up as parameters in my graph window now, right? So I have PCA1 versus PCA2, right? PCA1, let's say, versus PCA3, right? So I can change those here on the axis. And you can see that as we start to go down the PCAs, right, they're becoming more ball-like, kind of look like smears. And that's just because the variance is going down, right? And so that's part of the reason why you might not need all of your principal components. So just keep that in mind. Okay, so now I have all of my principal components. So I'm going to feed those components now into my t-SNE. So let's launch this one more time. We're going to pick our quality cells, dimensionally reduce. We're going to go with the t-SNE. Right, but this time, instead of the genes, I'm going to give it parameters. So we'll pick the parameter tab, right, and just filter out. I just want my PCA run, right? So here's all of my PCA components. Add those in, right? Select. Okay, and I'm going to go ahead and run this, and then I'll come back to what all these dialogues mean. I just really want to get this going because it can take a minute or two. Well, let's just go ahead and get that going. Okay, so let's launch that again so we can talk about it. So t-SNE, so what do all these different knobs mean? So here again, you can name the t-SNE. I'd recommend doing if you're going to try it a couple different times, right? We picked the parameters, we picked our PCA parameters. Right, so if you pick the auto-SNE approach, right, that's going to kind of automate the selection of the first two knobs here, right? It's going to optimize those based on your data set, right? Or I should say the first and last knob, right? So if you pick the auto-SNE, it's going to kind of automate the selection of how many iterations and your learning rate based on the size of your data, right? So iterations is how many times the algorithm runs to achieve the embedding, right? And so with this configuration, it's going to kind of adjust that automatically and optimize it for you, right? So if you want to know more about the details of that, there's the accompanying publication here. If you go with the manual approach, then you can manually tune these knobs if you'd like to. The perplexity is just going to help with the resolution of the islands, right? So if you have a higher number, your islands are going to look different than if they were a lower number, right? So you can adjust that to kind of achieve a different kind of embedding if that suits your needs. And then learning algorithms, right? So the exact, as the name would imply, is probably going to be a little more exact, might take a little bit longer to calculate. The annoy is just an improvement on speed because it uses approximations rather than exact calculations. Same for the gradient descent. Barnes-Hutt's the original. FIT-SNE or the FFT, fast Fourier transform interpolation, again, another speed improvement. Okay, so if you want the fastest embedding of your t-SNE, then go ahead and pick the approximate and the FFT. Otherwise, you can stick with the defaults, which are the exact and the Barnes-Hutt. So those are the knobs for running the t-SNE, right? So we've already run it, so I'm not going to run it again. Okay, so then here's our t-SNE, right? So this is all of our data compressed into a two-dimensional space, right? We have t-SNE 1, t-SNE 2. So we can already start to do some visualizations with our dimensionality reduction. right? So if we go back to our options here, and instead of pseudo color, right, we ask for a heat map statistic. So that's going to unlock kind of a third parameter to visualize on top of our dimensionally reduced space, right? So if we're looking at t-SNE 1, t-SNE 2, if we go ahead and unlock this third parameter, maybe I want to see where this gene is, DDX11L1, right? So I don't see much of it there. But where's all the rest of my genes, right? This isn't all of them. So this is going to just open a side window, right, where you can access now all of those genes. Okay? So maybe I'm only interested in my highly dispersed genes, right? So something like ISG15, right? I can see a couple green dots up there. Maybe IFI16. We can also change this to large dots, right? Maybe that'll help us see a little bit better. And let's go back to our genes and gene sets. So highly dispersed genes, let's look at something else, right? So I can just see where those genes are falling kind of on the dimensionally reduced space there, right? Maybe we'll look at something that's a little more abundant, like maybe like CD8, right? So I can see a lot of CD8 transcripts falling in this island here. So maybe I've got CD8 or NK cells falling in this region. Yeah. All right, so this is one way you can kind of do it manually, but if you have a bunch of transcripts that you have in mind, we can also use color mapping within the layout editor to do that. So if we go ahead and go back to the layout editor, all right, let's make a new layout with the plus button. Now let's go ahead and drag in our quality cells, right? There's our t-SNE plot. So let's right click and do make multigraph overlays, multigraph color mapping. Right, and maybe I want to focus on just my highly dispersed genes, and maybe I have an interest in, you know, granzyme genes, right? So I'm going to go ahead and pick these granzyme genes and hit OK. Right, and there's that same kind of visualization, right? I can see where those granzyme transcripts are falling in my t-SNE space, and I can see that, you know, a lot of them are falling kind of in the same island there, right? So if later I find that I'm becoming really interested in granzyme and all of the genes in the granzyme family, maybe that's where a static gene set would come in handy, right? So here we've had these analytic gene sets that we've made off of gates, but we could also make a new static gene set, right? Open up that editor, and I'm going to call this granzyme. Alrighty. And then off my highly dispersed genes, I'm going to look for just the highly dispersed granzyme ones. And save, right? And so now I've got a gene set of granzyme, all of my granzyme genes. Okay. All right. So I think that's about all I wanted to cover for the dimensionality reduction. So let's go ahead and talk about clustering. That way we can start kind of wrapping up here. So now that we've done our dimensionality reduction, maybe we want some hard, you know, populations to look at and compare within our sample. So let's go ahead and do that. So we'll hit our quality cells, go ahead and launch the, you could launch the clustering platform, right? So this is going to ask you if you want to do the k-means or the k-nearest neighbors. But I'm actually going to go ahead and use a plugin. I'm going to use Phenograph just because it's a little more unsupervised, doesn't require as much input from the user. So this is just going to take a second to open. All right, so here's my Phenograph plugin, right? So it's going to ask me, what do I want to run the clustering on? Okay, so some people choose to use, you know, for instance, you could use all of your highly dispersed genes, right? In this case. But you can also feed parameters, right? So if you don't want to do your highly dispersed genes, you want to just pre-digest the data and use your principal components, you can do that too. I feel like the jury's still kind of out on which one is best, but you could certainly do both and see what gives you the best resolution of your clusters. Okay. So I'm going to go ahead and feed it the principal components just because it'll be a little bit faster. If you ever have an opportunity with plugins to save the R script, I always recommend you do, because if you have issues, then tech support will ask you for these files so they can see, you know, what went wrong in the code. Okay. Right, so I'm going to go ahead and check that box. K value, I would start somewhere between 30 and 50, but similarly adjusting those numbers is going to give you different numbers of clusters. Right, so I'm going to hit OK. So while Phenograph is running, I think I'm going to go ahead and just show you, where you can access these plugins. So if you go to the FlowJo Exchange, on our FlowJo website, you'll just go over to Downloads, FlowJo Exchange Plugins. All right, and this is where all of our plugins are, right? You can see these cards, which give you a little bit of information about each of the plugins, which software they're available in, whether FlowJo or SeqGeek. And you can apply some filters here, right? So if we're only interested in SeqGeek at the moment, I'll get rid of all the FlowJo ones. Maybe I'm not interested in quality control. Then you can start to kind of narrow down the ones that might be of interest to you. And if you just hit one of the cards, like Phenograph, for instance, right? This is where I got Phenograph from. You just hit that card, right? You can get a description of what it is, all of our previous versions. And when you download the plugin, it'll come in a zipped file, right? You'll just put the jar file on your plugins. And it'll sometimes have a kind of an info sheet on how to execute the plugin and any extra installation instructions. So I highly recommend reading through that. All right, so our Phenograph finished running, right? So as I warned you, right, it doesn't give you much to look at, right? It just shows you all of your clusters, right, that it found. So in this case, it found 28 different clusters, right? So that's what it found for me. So it tells me that each of these are unique in some way, and that's why they've been grouped together. But we, ultimately don't really know what they are or how they're different from one another, at least yet. Okay? So if we want to just do some visualizations, right? Just play around in the layout editor. We can similarly, drag in that plot of our quality cells. Let's go ahead and make that a little bigger so we can see it. Oops, my mouse seems to have frozen. There we go. Actually, I'm just gonna delete this and do it again. There we go. And I'll just zoom in. That'll be easier. Okay. And let's go ahead and change this axis. So if I hit properties, right, it can come in and change the axis to my t-SNE 1 and my t-SNE 2. Right, so you'll notice this window looks a lot like our graph window, right? We have all of the same knobs, right? We can change what's on the axis. We can change what it looks like, the colors, right? Hit OK. So there's my t-SNE run, right? And maybe I want to overlay my clusters on top. So if I just select my clusters, drag them right onto my t-SNE plot, right? It's probably going to take a second to populate because that's a lot of data, but you can see that I can visualize now all of my clusters together on the same dimensionally reduced space, right? My eye is going over to this yellow cluster because if you recall, that's where I saw a lot of granzyme, right? So maybe I'm getting clues that this island is going to be some kind of a cytolytic cell, or some kind of an activated cell, right? So maybe I'd want to focus in on that yellow cluster, right? Two shades of yellow here, but it looks like it's maybe cluster number 13. Okay? And just know that in these overlays, as I mentioned in the talk, they are completely customizable, right? So if I decide that I want to make my quality cells a different color, maybe I want to make them gray, you can go ahead and change that color. And you can also change the layers, right? So each of these is a dot plot right now, but if I select an entire layer, right, maybe my base layer, hit that, go to properties, specify, right, maybe I want that to be more of a contour plot, right, so it stands out more. You can do something like that and those changes will apply. Okay. And you can also change dot size to help with your resolution, and you can relabel axes if you'd like to, all within that dialog window. Okay, so to wrap up, let's go ahead and talk about differential gene expression, right? So I talked about how I might be interested in this island because it kind of looked like it coincided with a lot of my granzyme expression, right? So let's see, maybe I want to know how this island or this cluster in particular, so cluster 13, is different from everything else, right? So I can do that in a few different ways. One is to, directly compare cluster 13 to something else, right? So I could say, want to pick cluster 13 and I want to change to gene view. And now I can pick populations. I want cluster 13. And I want to compare it to what, let's just say cluster 12, right? And so now I can see all of the genes that kind of classify cluster 13 over cluster, what was that, 14 or cluster 12, sorry, over cluster 12, right? But maybe I want to compare cluster 13 to everything, right? I don't want to go and do this manually for each of my clusters. So that's where Boolean gating can come in handy, right? So cluster 13, I'm going to go over to my workspace. Where is my, I remember now, I think it came off. Yep, so if you're ever missing a button from your ribbon, sometimes they can be accidentally dragged off, right? You can always open your ribbon and bring it back into your ribbon. So that's what I'm going to do now. So I'll put it in my workspace tab, Boolean. Let's go ahead and drag that in, right? And now I've got my Boolean gate options here. So for cluster 13, I want a not gate, right? So I want SeqGeek to make me a gate that includes all of my clusters except cluster 13, right? So if I do that, usually puts it towards the bottom, right? So you can see how it's 86% of my events, right? Because it's all of my clusters except cluster 13, right? And so now I can do the same thing, but I have an actual like bulk population to compare it to, right? So let's go ahead and pick cluster 13 again, but this time on this axis, I'm going to put not cluster 13. Right, so cluster 13 and not cluster 13. So I could certainly make a gate here and say, clustered in cluster 13, but I don't really know the directionality, right? I just know that these genes are unique to cluster 13 compared to the others. But what if I want directionality for whether it's upregulated or downregulated? Okay, so this is where we hit the differential gene expression module. Okay, so basically it's going to make volcano plots for us. So let's launch that. And actually, let's change this to our highly dispersed genes. Okay, so a little less to work with now. So highly dispersed genes, these are unique to cluster 13. These are unique to everything else that's not cluster 13, right? But I want to know directionality. I want some stats. Go ahead and launch my volcano plot, right? And so now, a little hard to see. Let's make those dots a little bigger. Okay, so now I can see fold change, right? So whether those genes are upregulated in cluster 13, right? So cluster 13 is on my x-axis, so it's going to be the one chosen for the volcano plot. So up in cluster 13 or down in cluster 13, right? This is a negative fold change, meaning it's going down. And we have kind of an inverted p-value axis here, right? So p-value of 0.05, it gets lower as you go higher. This is an inverted axis. Right, so let's say I want the genes that are at least 2 times upregulated, right, in my cluster 13 and that are significant, right? So I'm going to just draw that gate, which is, again, it's going to make a gene set because we're in gene view. And I'm going to call this 2 times up in 13, right? Or we'll say cluster 13. All right, and here's my gene set, right? So what are those transcripts, right? If I pick that gene set, I can see what's in there, right? So this is an analytical gene set, something that I can't edit. It was made off of a gate, right? And if I adjust the gate, then it'll update, right? But the list itself is not editable, right? So I can sort by fold change, right? Or maybe sort by p-value instead, right? And I can see some of these genes that are characterizing that cluster. Right. So then if I say, done, well, what can I, do with that gene set other than inspect those genes manually to see, what makes cluster 13 unique? I can take that, export that cluster, right? Let's just do a CSV format. Oh, opened in another window. Here we go. I'm going to just put that in my desktop quickly. Right. And if we decided to, ask what's in that, we could go to a web tool like this is probably one of my favorites, Panther, but there are others, and I can show you also how to do it within SeqGeek itself. But I'll go ahead and just give it the genes that I exported, right? 2 times up in cluster 13. Open that up, tell it it's human. I like to do the visual, so classification with pie chart. Let's hit submit. Oh, whoops, session expired. Let's try. Must have had this open earlier. All right, there we go. Okay, so then it's just going to give me some insight onto where those genes, like what kinds of pathways they fall into, right? So I've got some based on molecular function. Maybe I want to say pathway instead of molecular function. No hit for Panther pathway here. Let's go back. All right, let's try biological process instead. Okay, so we've got a different bunch of biological processes here. So there's a bunch of different ontology ways that the genes can get classified, right? So you can just mouse through them and see what interests you. So we'll say biological process, right? So I've got a lot of hits for this blue color. So let's kind of zone in on that. Cellular process. I'm gonna just take a second to load. Right, and it's going to tell me which were those genes that showed up for cellular processing, right? It looks like I've got some transcript proteins, some transcription factors, right? And so then you can get some information about these genes as well as what kinds of pathways they fall into, right? So if you wanted to pick a different pathway, you could just, so here I've got some hits for metabolic processes, right? I'm getting some more the same genes. So it's just a way you can explore those gene sets and learn, what might be in them. And there are other platforms that way. You can also, hit your cluster, do an enrichment test, right? And if you have gene sets you've imported, some of them are included in the demo data, you can import that gene set, run it, and it'll give you a similar analysis. All right, so I think that is all that I wanted to cover today. I will quickly show the ISLR plugin just to show that it can help automate this gene set formation. So I'll go ahead and just open a new workspace to show you what happens when that launches. Let's go ahead and close some of that stuff. All right. So, I showed the process of how you can look for differential gene expression in your clusters, right? Whether it's up in cluster 13 or down. But if you use the ISLR plugin, it can help you automate that process, right? So once you execute that plugin, it's going to automatically create those gene sets for you and make them organized based on whether it's up or down in a certain cluster. Yeah. Right, so you can see that this was the end product from my ISLR. So if I had just gone to, in the case of my other workspace, if I go back to it, if I'd gone here and gone to my workspace, gone to my plugins, launched ISLR, right, asked it to do differential expression, right, and I would pick, which genes I wanted to go into it, pick my clustering category, and do a fold change cutoff, right? Everything maybe over 2 folds, everything over a certain p-value, right? And so then once that's done running, which might take a few minutes, then it's going to just give me everything I just did manually, but for all of my clusters, right? So here it's going to tell me all of the gene sets that are down-regulated across each of my clusters, as well as all the ones that are up-regulated across each of my clusters, okay? So just a quicker way to kind of automate, what we just went through with the volcano plots. Okay? And then in terms of pipelines, we do also have automated pipelines in the form of plugins, which can basically do everything from quality control to clustering to dimensionality reduction, everything of that sort. So I can also show quickly, what something like that would look like. So if I was going to run the plugin Seurat, right, so I would choose that plugin. Let's go ahead and open a new workspace so we can look at it. Just gonna look for it here. Okay, get that open. Right, and while that's loading, I'll just say again, what's going to happen in the Seurat pipeline is you're going to end up with a lot of the things that we've just done manually, right? We did our dimensionality reduction, we did our clustering, and we did differential gene expression using the kind of volcano plots module. So you can see here, a lot of that has already been done, right? So I have the Seurat clusters that were made for me. I had my gene sets for downregulated or upregulated in each cluster. And it also does some visuals for you, right? So it's going to already go ahead and make a heat map for you. And it's going to show it by each of the clusters. Let's see if I can scroll over a bit. Right, and it can also do these kind of rain cloud plots showing you your RNA features. Right, I could do some dimensionality reduction. See if I can scroll down to the ones that I want to show. All right, there we go. So there's some dimensionality reduction with all of the clusters overlaid on top of the dimensionally reduced space. So it also saves you some time with having to create some of these visuals. Okay, I think that is all that I wanted to show today. I see that we're a little over time. Just a reminder that please feel free to reach out if you have any questions, any follow-up questions. I look forward to seeing you all at the next SeqGeek webinar. And enjoy the rest of your day.